friends, this is Dr. Varsha. Welcome to another video lecture from Botany Varsha. We have already studied difference between eukaryotic and prokaryotic DNA replication, and today we shall study process of DNA replication. So let's start. The process by which the parent DNA molecule gives rise to two exactly similar sister DNA molecule is called DNA replication. For this process, many things are required like different type of enzymes, nucleotide pool, etc. So first thing which is very much important, that is nucleotide pool. During the process of DNA replication, 4 different kinds of nucleotides are required. These are deoxyadenosine triphosphate. This is also called DATP. Deoxyguanosine triphosphate, DGTP. Deoxycytidine triphosphate, DCTP. And deoxythymidine triphosphate. This is also called DTTP. These nucleotides are formed from their monophosphates by the process of phosphorylation, catalyzed by enzyme phosphorylase. They are found freely inside the nucleoplasm, forming the nucleotide pool. Next thing is enzymes. Different type of enzymes are required during this process. DNA replication is highly enzyme dependent process. There are many enzymes involved in the DNA replication, and DNA polymerase is the main enzyme during this process. So DNA dependent DNA polymerase, it helps in the polymerization and regularize the whole process of DNA replication. With the support of other enzymes, deoxyribonucleoside triphosphates are substrate as well as the energy provider for the replication process. In prokaryotes, DNA polymerase is of 3 types. DNA polymerase 1, this is DNA polymerase 1 in prokaryotes. It is responsible for DNA repairing. And second one is DNA polymerase 2. DNA polymerase 2 is responsible for primer extension and proofreading. And this is DNA polymerase 3. DNA polymerase 3 is responsible for DNA replication. And in eukaryotic cell, DNA polymerase is of 5 types. DNA polymerase alpha, which is responsible for primer formation and initiation. And DNA polymerase beta, which is responsible for DNA repairing. DNA polymerase gamma, it performs the function of DNA replication in mitochondria. DNA polymerase delta, this is DNA polymerase delta, and it performs actual DNA replication in eukaryotes. And DNA polymerase epsilon, it performs DNA replication during the budding of yeast. So next enzymes, some other enzymes are, this is topoisomerases. These are concerned with the breaking and resealing of DNA strands to relieve the tension of the coiling, and it keeps DNA untangled. So function of topoisomerase is to relieve the tension of the coiling. And next one is DNA helicase. DNA helicase, it helps in unzipping the DNA strands by breaking the hydrogen bonds between the nitrogenous bases. Thus, it helps in the formation of replication bubble and replication fork. And next is DNA ligase. DNA ligase present in both prokaryotes and eukaryotes. Ligase is the enzyme which glues or join the discontinuous DNA strands. Next is a DNA primase. This enzyme help in the synthesis of RNA primer, and which is complementary to the DNA template strands and which provide 3' hydroxyl group to the DNA polymerase delta, so that DNA polymerase delta can perform the chain formation. And next is SSBP. SSBP is single stranded binding proteins, and which binds to the DNA and protect it from forming secondary structure. Both DNA strands get separated from each other. So these are the things which are required for DNA replication. Next is the process mechanism of DNA replication in eukaryotes. So first function is initiation, and DNA replication demands a high degree of accuracy because even a minute mistake would result in mutations. For the replication to begin, there is a particular reason that is called origin of replication. This is the point where the replication originates, and the replication begins with the spotting of the origin and followed by the unwinding of the two strands and unzipping of DNA strands in its entire length is unfeasible due to high energy input. So hence first a replication bubble or replication fork is created by helicase, and by breaking the hydrogen bond between the nitrogenous bases, and the strands unbind. Now topoisomerase release stress and keep DNA untangled. So by this way initiation start in eukaryotes. And next second point is activation of deoxyribonucleotides. Deoxyribonucleotides occurs freely in the nucleoplasm. They are of 4 types: DAMP, DGMP, DCMP, and DTMP. Means deoxy in the form of monophosphates, means they are found in the form of monophosphate in the starting, and they are phosphorylated and convert into active form, in which 3 phosphate groups are found. Means in active form monophosphate convert into triphosphate too. DAMP convert into DATP, DGMP convert into DGTP, DCMP convert into DCTP, and DTMP convert into DTTP, thymidine triphosphate. So after activation of deoxyribonucleotides, exposure of DNA strands, and helicase enzyme unwind the DNA double helical structure and separate the 2 strands. Topological stress is released by topoisomerase enzyme, and energy is provided by ATP. The separated strands are stabilized by SSBP, single stranded binding protein. SSBP bind to DNA strands as tetramer, and the 2 DNA strands open for replication. DNA does not open in one stretch. It opens slowly and gives the appearance of replication bubble and Y-shaped replication fork. And next is this is DNA strand, and in which different nitrogenous bases are attached, and now topoisomerase attached to the DNA strand, which release the stress from the DNA strand, and now helicase enzyme break the hydrogen bonds between the nitrogenous bases, and Y-shape replication fork is produced. And now the single-stranded binding proteins are attached to the both the separate strands in the form of tetramer, and topoisomerase release the stress from the DNA strands. And DNA strand is antiparallel, which is always found in 5 prime, 3 prime, and 3 prime, 5 prime direction. And SSBP, or single-stranded binding proteins, is attached to the both separate strands in the form of tetramer. So these SSBP protein stabilize both the DNA strands, so they cannot attach again with each other. And next point is primer formation. RNA primer is essential for DNA synthesis. It is the small strand of 4 to 12 nucleotide long RNA, and this is synthesized at the free 3 prime hydroxyl end of DNA, which is available in the region of origin of replication on the other strand. It is present in the replication fork. So it is produced on the different position of the 2 DNA strands. And after the formation of primer, nucleotide chain formation start with the help of DNA polymerase third in prokaryotes and DNA polymerase delta in eukaryotes. After the start of nucleotide chain, RNA primer is removed and the gap filled with the help of DNA polymerase 1 or DNA polymerase delta. Now, this is a parent DNA template, and which is in 3 prime 5 prime direction. So for the activity of DNA polymerase, 3 prime end with hydroxyl group is required for base pairing, and which is provided by primer. So this is primer. And primer is short stretches of RNA nucleotide, which is about 10 to 12 base pair long, synthesized by primase enzyme. If 3 prime OH, 3 prime hydroxyl group is not available, so base pairing and new strand formation is not possible. So this is DNA polymerase, which can only attach on the 3 prime hydroxyl group. So then now when the DNA polymerase start chain formation, and new template is produced on the parent template, and it is always found in 5 prime 3 prime direction. This is primase synthesize short stretches of RNA nucleotides, providing a 3 prime hydroxyl group to which DNA polymerase can add DNA nucleotides. Next point is base pairing, and the two separated DNA strands in the replication fork function as template deoxy ribonucleotide triphosphate come to lie opposite to the nitrogenous bases of exposed DNA template. And pairing of nitrogenous bases is always specific, in which adenine, which is purine, pairs with thymine, and that is pyrimidine, and guanine, which is purine, again pairs with cytosine. This is known as complementary base pairs, and in which deoxythymidine triphosphate convert into deoxythymidine monophosphate, which arranged opposite to adenine, and again deoxycytidine triphosphate convert into deoxycytidine monophosphate, and which arranged opposite to guanine, and deoxyadenine triphosphate convert into deoxyadenine monophosphate, which arrange opposite to thymine, and deoxyguanosine triphosphate convert into deoxyguanosine monophosphate, which arrange opposite to cytosine. And triphosphate convert into monophosphates, and phosphate which release which again react with water and produce energy. So this energy is utilized in the base pairing during hydrogen bonding. Next point is, this is a parent DNA template on which topoisomerase is attached. All the nitrogenous bases are attached, in which cytosine pairs with guanine and adenine pairs with thymine. Now topoisomerase enzyme release the stress from the parent DNA and get both the strands separated, and now helicase and breaks the hydrogen bonds between the nitrogenous bases, and both the strands get separated from each other. And now these are two strands which get separated from each other. These are single-stranded binding proteins which are found in the form of tetramer, and free nucleotides are found in the nucleoplasm. And now DNA replication start with the addition of the primer. This is a primer, and this primer is attached. First primer is required, then which provide 3' hydroxyl group to the DNA polymerase. Then the DNA polymerase attach, and now base pairing start. Adenine opposite to the adenine thymine, and opposite to cytosine guanine, again opposite to the guanine cytosine, and opposite to adenine thymine. So now this way a small strand is produced, and DNA replication is always found in 5', 3' direction. So new strand is always produced in 5' direction. For the activity of DNA polymerase, 3' and with hydroxyl group is required for base pairing. And a primer is very small stretch, around 10 to 12 base pairs, which is first attached, then the DNA polymerase is attached. And during the process, and DNA polymerase third is required in prokaryotes, and DNA polymerase delta is required in the case of eukaryotes. And then in the nucleoplasm, so many free nucleotides are found which get attached during the process. And now again primer is attached, then DNA polymerase is attached, and base pairing start. Then DNA polymerase start base pairing, again opposite to guanosine cytosine pairs, adenine pairs with thymine, thymine with adenine, and adenine again with thymine pairs with adenine, and adenine again with thymine. So next new small strand again produced in 5', 3' direction. So by this way small fragments are produced during this process. These small fragments are called Okazaki fragments. And in the other side, on the other side, only one primer is required, and then primer provide 3' hydroxyl end to the DNA polymerase, and DNA polymerase start base pairing. And now the base pairing start again in the same way, and purines with pyrimidines, and guanosine attached with cytidine, thymine with adenine, adenine with thymine, guanosine with cytosine. So by this way base pairing is done during this process, and new strand is produced. In this, by this way, new strand is produced. This strand is called leading strand. In this process, only one primer is required, and because there is continuous DNA replication, and after some time, this gap which is produced between the Okazaki fragments, this is filled by DNA ligase, and again this discontinuous small fragments convert into continuous fragment. This is called lagging strands, and in this by this way, on the one strand discontinuous DNA replication is found, and on the other strand continuous DNA replication is found. This is called semi-discontinuous DNA replication. And now next is this is parental DNA template strand. They are antiparallel to each other. This is now the topoisomerase, which is concerned with the breaking and resealing of the DNA strand to relieve the tension and coiling and keep the DNA untangled. Now the helicase separate the parent DNA strands. DNA replication always start in 5' 3' direction. On one strand, small fragments are produced. These small fragments are called Okazaki fragments. Here DNA replication is discontinuous, called lagging strand. This is called lagging strand, and the gap which is produced between the different small fragments, these are filled with DNA ligase. But on the other hand, only one RNA primer is produced, then DNA polymerase starts synthesizing new strand, and this replication is continuous, and this is called leading strand. Okay, so DNA replication is discontinuous on lagging strands, and on which new primer must be generated at the beginning of each Okazaki fragment. And on the other strand, on this strand where the replication is continuous, called leading strand, here primer is required only at the 5' end of the newly synthesized DNA strand. So by this way DNA replication takes place. And next is chain formation. It is to be noted that elongation is unidirectional. As the strands are separated, the polymerase enzyme starts synthesizing the complementary sequence in each of the strand, and the parental strands will act as a template and for newly synthesizing daughter strands. And elongation is always unidirectional. DNA is always polymerized only in the 5' to 3' direction. Therefore, in one strand, on the one strand called leading strand, where the replication is continuous, and on the other strand replication is discontinuous, and this strand is called small strands are produced. These are called Okazaki fragments, and this after the joining by the DNA ligase, this is called lagging strand. And now at the completion of the synthesis, RNA primer is removed by DNA polymerase beta, and DNA ligase seals the breaks between the Okazaki fragments as well as around the primers to form continuous strands, and the DNA replication is continuous over one strand and discontinuous over the other strand. It is called semi-discontinuous DNA replication. And now next point is proofreading and DNA repairing. This system comprises removal of mismatched nucleotides during replication and insertion of correct complementary nucleotides in their places. Sometimes DNA bases or DNA segments get damaged in DNA repairing system. These damaged portions are replaced with normal DNA segments, and proofreading and DNA repairing is done by activity of DNA polymerase 1. And now next step is DNA double helix formation, and helix formation follows soon after the process of replication is completed in a segment of DNA. After that, nucleosomes are produced. And now significance of eukaryotic DNA replication. DNA replication is a fundamental genetic process that is essential for cell growth and cell division. And DNA replication involves the generation of new molecules of nucleic acid. DNA crucial for life. And next, DNA replication is important for properly regulating the growth and division of the cell, and it conserves the entire genome for the next generation. So this DNA replication is very important process. Thanks for watching. Stay connected for new lecture from genetics. Thank you. Have a nice day.